Let's continue with the structure of fructose, that is the cyclic structure of fructose. The chemical formula for the fructose is C6H12O6. The other name for the fructose which is used, it is fruit sugar. It is fruit sugar. Okay. How it is obtained? When cane sugar, that is sucrose, it is been hydrolyzed, it is undergoing hydrolysis in the presence of dilute sulfuric acid, we will obtain two C6H12O6 molecules. And in this, the one is glucose and the other one is fructose. Okay. So this is the preparation method for the fructose. Now, this fructose is also the solution having equal molecules of D-glucose and D-fructose is called invert sugar. If equal amount of both these compound, that is D-glucose and D-fructose is present in the reaction, then that particular sugar is termed as invert sugar. Now the structure of fructose. So the structure of fructose is 1, 2, 3, 4, 5 and 6. 4, 5, 6, 6 carbon atoms. At the topmost and the bottommost carbon atom will be having CH2OH. The middle carbon it will be having a ketone HOH, OHH and OHH. So this is the structure for the fructose. Now this is the open structure for the fructose. Okay, D fructose. The cyclic structure for the fructose will be here CH2OH, COH, 3, 4, 5, H, OH, third carbon atom, fourth carbon atom and fifth carbon atom. OH will be on the right hand side and H will be on the left hand side. CH2. Here the bonding of oxygen will be with the carbon atom of the last carbon atom. While in the case of D-glucose, we have seen that it was with the fifth carbon atom. So here 1, 2, 3, 4, 5, 6. So here the second carbon atom where the ketone group was present, it was forming the hemiacetals with the last carbon atom here of the fructose. Okay, to form the hemiacetal, and this particular compound is termed as D alpha D fructose. What is happening here? Here the CH2OH, C double bond O, CHOH, COH, H, COH, H, C, 1, 2, 3, 4, 5, 6, H2OH. Okay. So what will happen here? This double bond of the oxygen, carbon with oxygen, here the carbonyl group will break its bond. Okay. And one hydrogen from this carbon atom, it will attaches to this carbocation which is found out here. And the oxygen here, okay, the oxygen here which is present, okay, the oxygen here which is present in the OH group of the last carbon atom, it will just attack on the carbocation which is formed out here after the bond breaking between this carbon and oxygen. And the H here, it will get joined to this O here, which will attain the negative. So here two different structures can be formed. One is this alpha one, okay, alpha fructose, and the second is it is been distinguished by the position of the CH2OH. When the CH2OH is positioned in the right hand side, it will be termed as the it will be termed as the beta one, okay, the beta fructose. While if it is been attached in the left hand side, it will be termed as the alpha fructose. this. Okay, so here it will be beta fructose when the CH2OH is attached in the right hand side. 2, 3, 4, 5, 6, 7. Oh, one carbon atom I have attached more. So here it will be CH2 and the oxygen will be bonded with this last carbon atom like this. H. So this is the structure for your beta fructose. Beta fructose. Okay. So for the Haworth projection of these two compounds, that is the alpha D fructose and beta D fructose, we can use the carbon bonded with oxygen, then with carbon, 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 carbon. Okay, one, two, three, four, five. The six carbon here it will be CH2OH, down OH. Here it will be OH, H, H, OH, H, OH, and the last carbon atom it will be having one hydrogen above and one hydrogen below. Okay, this is the alpha D fructose or fructose, or the other name will be alpha D fructopyranose, fructopyranose. And for the structure for the CH2OH, carbon, carbon, carbon. Okay, six carbon has been attached with each other. Here the CH2OH will be at the downside and OH will be on the upper side here. OH, H, H, OH, H at the downside OH, and here one H above and one H below. So this is the structure for your beta D fructose or beta D fructopyranose. Fructopyranose. This is the Haworth projection. Haworth projection for the alpha fructose and beta fructose. In the free state, D fructose it exists in the six-membered ring or as a pyranose ring. However, in the combined state as a component of disaccharide, okay, when it is present as a disaccharide, it is existing in the form of furanose form. Furanose form when present as a monomer of disaccharide. Saccharides, disaccharides. Okay, so here the structure will be like this, double bond. Okay, this is termed as furan. So here CH2 OH C C C C C H2 OH and one H here, and the carbon atom, fifth carbon atom will be contained out with the second carbon atom where double bond O was there, and the structure will be of the compound will be like this. So this is the D fructofuranose. D fructofuranose. And if the CH2OH is present in the right hand side CH2 OH, OH, 1 2 3, 1 2 3 4 5 6. Fifth carbon HOH, CH2OH, here OH, third carbon, sorry, third carbon we require this. So here it will be HOH, it will not be here. Here it will be OH, H, HOH, and the carbon atom here it is attached with the oxygen. This is the beta D fructofuranose. Fructofuranose structure. Okay. So this is the Hayward structure for the fructofuranose. So here we can represent the structure CH2OH at the bottom OH, OH, H, H, OH, H, OH, H at the bottom OH, and at last here O. Similarly, this is the alpha fructofuranose, and for the beta fructofuranose it will be C, OH, CH2OH, OH, H, HOH, H. Okay, this will be the structure for the beta fructofuranose. CH2OH at last here, CH2OH at last here. Okay, so this is the structure for the furanose, fructofuranose, that is alpha fructofuranose, alpha fructofuranose, and this is the beta fructofuranose. Now let's start the next topic, that is the proteins for the biomolecules. So what are proteins? Proteins are higher molecular mass, complex biomolecules, complex biomolecules of amino acids, present in living cells, okay. So the name protein is derived from the Greek word proteos, which means prime importance. This particular word proteos means prime importance. So the chemicals of prime importance, okay, the chemicals of prime importance are the proteins. So what are proteins? What are the different types of proteins? Where we can find the proteins? So proteins can be found out in the enzymes. The enzymes are made up of protein. Okay, the hormones present in our body, they are also nothing other than the protein. Then the antibodies, they are the proteins. Transport proteins are also present in our body. Transport proteins, structural proteins, structural proteins, contractile proteins. So all these are the examples of the types of proteins which are present in our body. So how, what are proteins in the chemical way? If we are going to see what are proteins, so proteins are actually polymers. Okay, so on hydrolysis of protein, it will break up into the peptides. Okay, it will break up into the peptides. Then further, when peptides are hydrolyzed or hydrolysis of peptides is taken place, then formation of amino acids take place. Amino acids takes place. So thus we can say that chemically proteins are condensates. Proteins are condensation polymers. Condensation polymers. Condensation polymers in which monomers units are alpha amino acids. Now let's talk about the amino acids then, because the main units thing which forms the proteins, they are nothing other than the amino acids. And what are the amino acids? So amino acids are compounds. They are the compounds containing containing both amino group and the carboxyl group. And the carboxyl group. The general formula for the amino acids will be R. General formula for the amino acids will be R C H. One carboxyl group is there. Okay, one carboxyl group is there, and one amine group is there. Okay, in this particular compound we will see that one carboxyl group is present and one amino group is present, and this is the side alkyl chain. Side alkyl chain. Okay, so these are the amino acids. Now all the amino acids have chiral carbon atoms. Okay, generally except glycine, all other amino acids have chiral carbon structure and have two optically active isomers there. Okay, it will be present two optically active isomers. If I am taking the example NH2 COOH, the structure of the carboxylic sorry the structure for our amino acids, here it will be like this, and here the R will be connected at the back side of the carbon atom, central carbon atom. Okay, this is the chiral. So let's see the chiral compounds. So the chiral compound here, it will be COOH, RH. Okay, so these both are the isomers for the alpha amino acid. Okay. Now let's take example for the L-glyceraldehyde. So in the glyceraldehyde, this is the L-glyceraldehyde and L-amino acid. Okay, if we are taking example for the L-amino acid. So we will observe here that okay. So here we will observe that these both are L here. Okay. So here in the L-glyceraldehyde, we will see that the OH group is present at the left hand side, while in the case of the L-amino acid, okay, this is L-amino acid. In the case of L-amino acid, the NH2 group is present at the left hand side. Okay. Similarly, in the L-alanine, that is L-alanine, the NH2 group will be present at the left hand side. L-alanine. Okay. Now, how to name the amino acids? Although amino acids can be named using the IUPAC systems, they are generally known by their common names. Okay. So common names for this particular compounds are like that glycine, alanine. Okay. So for CH2, COOH, it will be glycine. That is capital G is used for its naming. CH3, COH, NH2. This is alanine. That is capital A is used for representing alanine. Okay. So these are the way to name the compounds of the amino acids. Now, types of amino acids. Types of amino acids. So amino acids can be classified into three types. The first is acidic amino acids. The second is basic amino acids, and the third is neutral amino acids. So in a compound, in an amino acid compound, where COOH group is more, that is the carboxyl group is more in number than NH2 group, then that particular amino acids will be termed as what? They will be termed as the acidic amino acids. Okay. And we can say that more than one COOH group is there and only one NH2 group is there, then they will be termed as the acidic amino acids. When NH2 group is more and only one COOH group is there, that is the carboxyl group is there, then the amino acid will be termed as the basic amino acid. And a neutral amino acid, where number of COOH group is equals to number of NH2 group. So that particular compound will be termed as what? They will be termed as the neutral amino acids. Okay, neutral amino acids. What are some of the properties of the amino acids? Properties of amino acids. So properties of amino acids in that are C, H, NH2 with a lone pair of electron and COOH. Okay, so here as we can see that both acidic and basic group are present in the same compound only. So intra compound reaction will take place. Okay, intra compound reaction will take place here, and the H ion of the COOH will get attached to this NH2 group here to form a positive charge and a negative charge here in the presence of this compound. Okay, so here this particular ion which has been found, in which both the positive and negative charge is present on an amino group, that is termed as the Zwitter ion. Okay, Zwitter ion. And this salt which is formed, the salt which is formed here, it is also termed as the internal salt. Okay, it is termed as the internal salt. So here the presence of CO minus indicates that there is a presence of basic group here, and NH3 plus shows that it is the presence of an acidic group here also. Okay, so both kind of nature, that is the acidic nature and the basic nature, will be possessed out by the amino acids. Okay, so these are the properties of the amino acids. Next is the isoelectronic behavior. Okay, isoelectronic behavior of the amino acid. So due to presence of positive and negative charge in the amino acids, we can see two different behaviors. Okay, two different behaviors for the amino acid, and that behavior is when H plus ion is been removed and OH is been taken out, then we will see that the compound will form, it will be like this, where a minus charge will be there, means more basic nature will be there. And if the H plus ion and OH minus ion will take place here, and CH, COOH, H will get attached here. So we will see that NH3 plus will be there. So here the acidic nature will increase out. Okay. So the pH or hydrogen ion concentration of the solution at which a particular amino acids do not migrate under the influence of an electric field is called isoelectronic point. Okay. The pH, the particular pH or hydrogen ion concentration of solution at which we will see that at which a particular amino acid does not migrates under the influence of an electric field is called isoelectric point. That particular point will be, that particular pH point will be termed as the isoelectric point. Okay. So for basic amino acids, that particular pH is 7.6 to 10.8. For acidic amino acids, it is between 5.5. The pH must be between 5.5 to 6.3. And for the neutral amino acids, it must be around, sorry, this is for the neutral amino acid. This is for the neutral. This is for basic amino acids. And for the acidic amino acids, it must be, the pH must be between 3.2 to 3.5. Okay, at this particular point we will achieve the maximum number of isoelectronic points for the different amino acids. Now let's move further and see the peptide bonds. Peptide bonds. So peptides, what are peptides? Peptides are compounds formed by the condensation of two or more same or different amino acids, alpha to be more precise, alpha amino acids. Okay, so here the formation of bond will take place when two or more amino acids are combined with each other. It can be same also, or it can be different also. For example, I am taking an example for the glycine, okay, which is reacting with the glycine, which is combining with the aniline, okay, which is combining with the aniline, alanine, sorry, not aniline, alanine, to form, and the condensation process will take, the condensation formation will take place here, and water will be released out here, and the formation of, okay, here I can represent this NH2 like this, and with a lone pair 1H and 1H. So here the OH, it will break out with the H ion, okay, the OH from the carboxylic acid, it will break out with the H from the NH2 group here, to form the bond between the NH and NH of the alanine and the OH of the glycine, to form CH of the glycine, to form the compound here. This bond, CONH bond, this bond is termed as the peptide linkage. This is termed as the peptide linkage or peptide bond. Okay, this linkage is termed as the peptide linkage or peptide bond, and this compound is termed as glycyl alanine, that is glyella or Ga, glyella or Ga. Okay. Now, what will happen in the polypeptide bond formation? So during polypeptide bond formation, we will see that thousand number of, okay, thousands and thousand number of these peptide bonds will be formed out there, and the formation will be the polypeptide. For example, I can take one example here: CH, CO, NH2. Okay, NH2 is here, NH2 with one R group. It is bonded with the NH, CH, CO, R group, to the last group here, that is NH. Here n numbers of compounds will be there in between: NH, CH, COOH, and R. Okay, so this linkage is termed as the polypeptide linkage. Okay, polypeptide linkage. So two ends you can see here. So this end is termed as the N terminal of the polypeptide, and this end is termed as the C terminal. Two different endpoints or two different terminal points are here. So the two different terminals are here termed as N terminal end and C terminal end. The structure of the polypeptide is written with the names of the amino acid as they appear from the left to right, starting from the N-terminal amino acid, and then the suffix "ine" is the name of the amino acid present in the "ile" group. That is, the glycine will convert into glycine will convert into glycyl, alanine will convert into alanyl, etc. Okay, so here the naming is completed like this one. Here we can take an example for the naming purpose: NH2, CH2, C double bond O, which is compounded with NH, CH, CH3, C double bond O, compounded with NH, CH2OH, C double bond O, OH. So the name here, it will be three different compounds is there. One is your glycine, Okay, glycine. The middle one is your La9, and the last one is your here serine. So the name of this compound will become glycyl alanine serine, which is a tripeptide compound. Tripeptide compound, and the abbreviated name will be gly La ser gly La ser. Okay, so this type of naming can be used for the naming of the polypeptide bonds. Now let's talk about the proteins. Okay, this was for the polypeptide bonds. Let's talk about the proteins. So proteins are the complex nitrogenous nitrogenous so proteins are the complex nitrogenous compounds, okay, which are essential for the growth of the maintenance and life. So how we can classify the proteins? Proteins can be classification of proteins. So proteins can be classified on the basis of their structure. So the first is the fibrous proteins. Fibrous proteins. And the second one is the globular proteins. Okay. So what are the fibrous protein and globular protein? So let's discuss about the fibrous proteins. Proteins consist of linear thread-like linear thread-like molecules which tends to lie side by side to form fibers are fibrous proteins. Fibrous proteins. Some of the example for the fibrous proteins can be keratin in skin, in skin, in the hair, in the nails, and in the wool also. The collagens in the tendons, the fibrons in the silk, the myosin in the muscles. So these are some of the examples of the fibrous proteins. Next, what is the globular proteins? The molecules are folded. The molecules of proteins are folded together into into compact units forming spheroidal shapes. Spheroidal shapes. Okay. So the polypeptide bonds here, the polypeptide bonds in globular proteins are held by hydrogen bond, are held by hydrogen bonds. But these forces are comparatively weak. They are comparatively weak. They are soluble in water, or acids or bases, or acids, bases, or salts solutions. Okay, while the proteins, that is the fibrous proteins, are insoluble in water. Okay, due to their presence of the strong peptide bonds between them. Now, in the next class, for today's class, I am ending the class here only. In the next class, we are going to see the proteins on the basis of their structure. Okay, on the basis of the structure of the proteins. What is the different structures of the protein? So, four structures of proteins are there: primary, secondary, tertiary, and the quaternary. Four different structures of the proteins are there. We are going to discuss about them. Okay. So, for today class, I am ending our class here only. We will meet in the next class. Thank you.